Welcome to a Taconic T-Onco webinar entitled Preclinical in vitro and in vivo evaluation of combined radiation therapy, given by Dr. Guillaume Sarn from our French oncology partner company, OncoDesign. Let me introduce today's speaker. Guillaume has been working for OncoDesign for 11 years as a study director and is now head of the Cellular Pharmacology Laboratory. Guillaume obtained his PhD from the University of Toulouse, France, in molecular biology and biotechnology, followed by a postdoc at the Roswell Park Cancer Institute on the characterization of new proteins involved in the quality control of mRNA. He is now the Cellular Pharmacology Director at OncoDesign, working closely with clients and study managers to direct in vitro and in vivo preclinical studies. We hope you enjoyed today's webinar. Guillaume?
Yeah, well, thank you, Maggie, for introducing me. And thank you for all attendees for their interest for this presentation. So I will present the preclinical in vitro and in vivo evaluation in combined radiation therapy that we perform in OncoDesign. So next slide. So first, the rationale of this presentation is well, to speak about radiation therapy, which is the most frequent tumor treatment just after surgery, and it's sometimes given as a primary therapy, but often is combined with surgery, also chemotherapy or hormonotherapy. The radiation damages protein, lipids, but the principal molecule that is targeted is DNA, directly or via formation of oxygen radicals. The goal is cell death via apoptosis or mitotic catastrophe if possible. When associated with chemotherapy, well, chemoradiation classically involves the use of cytotoxic agents, also targeting DNA, to reinforce the effect of radiations. But now new target therapies are designed to identify new molecules that are acting as radiosensitizer by inhibiting proteins that are involved in resistance of cancer cells to radiotherapy. And for this characterization of new molecules, you need new preclinical in vitro and in vivo experiments for selection and characterization of these new molecules. So how I will organize my presentation. So I will make first an overview of irradiation effect, the tumor-resistant and potential sensitizer treatments that are already existing, a presentation of the radiation facilities that we use, a method for preclinical in vivo irradiation experiments with examples that we perform in OncoDesign, and also irradiation experiments. So first, the radiotherapy effect. So it is caused by the ionizing radiations, which can be particles or electromagnetic rays, and these are directed against they cause lesions on protein, lipids, and what is more interested us and the therapy are DNA. So this effect can be direct on DNA, but also a lot is mediated by the radiolysis of oxygen and water through free radicals. When the lesion is performed, there are three main choices. The DNA is not repaired, and in this case it goes to necrotic cell death, also when it is associated with the loss of protein and lipid degradation, and then induced tissue pathology, which is not good. When the DNA is repaired, you can go, if there are too much mutations, then it can go to necrotic cell death, and also in apoptosis, which is the way that we prefer for therapy because it is cleaned by the immune system, and then the tumors are cleanly disappear. And also, it can induce non-lethal mutation, and this is another way for getting cancer, and also hereditary abnormalities. And in most of the cases, indeed, the repair is correct. So this is not what you want to. And so the cells naturally acquire resistance, and this resistance can be a reduction of oxygen that diminish the free radicals accumulation, and this is hypoxia, and also an increase of glutathione that are needed for regulation of these free radicals into the cells. Also, there is an increase of DNA repair via different. Here I put the key proteins that are targeted, the topo1, PARP, or ATM machinery, and also a diminution of apoptosis. And here I put 3 members that are current in therapy: EIP family, the TRAIL pathway, and the BCL2 family. Sorry. So there are different treatments. So first is to reduce hypoxia, and there are the classical method is to use a VEGF inhibitor that induce normalization and a normal vascularization that increase the oxygen level. There are also used, like I said, in chemotherapy, the different molecules that are linking to the DNA, like platinum, doxorubicin, or temozolomide, and induce additional damages. Also, molecules that are involved in the DNA repair by replacing the nucleotides, like gemcitabine, or decreasing the synthesis of nucleotides, like 5-fluorouracil, and diminution of different proteins that are involved in the reparation, like topo1, PARP, or ATM. There are also diminution of inhibitor of apoptosis via different inhibitors, and I put also here the common pathways that are also regulators of DNA repair machinery and apoptosis, and that can be used to increase the radiation effect. Here are the different systems that we use in oncodesign to perform the irradiation. So we have a linear particle accelerator, which emits electrons that are transformed into X photons, and this is used to irradiate the plates for in vitro assays and to irradiate some orthotopic tumor models. And there are also we also use a lot a system derived from a Curie therapy system, which use the iridium-192 element for irradiation, which emits gamma photons. And this is used for subcutaneous tumor models and some orthotopic tumor models also. So first tumor model that I can describe you is the orthotopic glioma U87MG tumor model. So this tumor is implanted by serotoxic injection into the brain of the rat, and here you can see on the MRI that you perform in oncodesign the tumor. And I place here the irradiation area in yellow, so you can see that only half of the brain is irradiated. Here and on this figure, I show how do we proceed. So we anesthetize the rats with ketamine xylazine and put it under the support, and we put a contention marks to orientate the tumor to the upside. And there is a plastic which is called a bolus that reduces the X-ray penetration to just irradiate the upper part of the brain. In these two figures, you can see here the survival and here the tumor volume after irradiation. So I put here, so we have the radiotherapy is performed 5 days consecutively at 2 grays per day. These are classical standard irradiation doses that are given in clinics. And here I report the temozolomide effect just as a control. This is not a combination. So you can see on the survival that temozolomide induced an increase of the survival of 152%, and this is directly correlated with a high reduction of tumor growth of 92%. If you look at radiotherapy, you can observe that there is no so much reduction of survival, but there is a significant reduction of tumor growth. And this phenotype is often observed with tumor with orthotopic tumors, where we can observe no difference in survival, but a difference in tumor growth. Now, another tumor model. So here is a PC3-MM2 orthotopic tumor model. So this is a human pancreatic prostatic tumor that is implanted onto the prostate of male rats. And you can irradiate, we irradiate only the pelvis of the rat, as indicated in this figure. And here you can see an increase of lifespan of 55%. So this time there is an efficiency of the irradiation per se to increase the survival of animals. And this is correlated with a T/C of about 65% at D21. Here in this slide I describe how do we proceed for subcutaneous tumor model. So here we take again the PC3-MM2 tumor model, and in this case we inject cells onto the flank of the animal, here a rat, but we also perform a mice injection. And we have, when we want to irradiate, so we anesthetize the animals, and we have two plates that are covering the tumor, and here and here, and here there are canals through where the catheter are containing the irradium, the source of irradiation is going, and then stop at different points to irradiate the tumor at the chosen dose. So this is what we do with our different subcutaneous tumor model. And in this case, for PC3-MM2, you can observe that there is on the subcutaneous irradiation, that there is an inhibition of tumor growth of 55%. Also, here I reported another tumor model, which is a syngeneic tumor model, 41, which is a mammary cancer from mice, which is induced orthotopically in the fat pad of mice. And in this case, in this experiment, we did only one irradiation at 8 and 15 grays per day, only one. And here you can see that a single irradiation at this high dose can induce a significant, moderate irradiation. And in this case, we protect the body of the mice with a lead. So here I put another subcutaneous model, which is FADU, and is a classical tumor model. It's a human head and neck tumor. And in this case, we irradiate like the other standard irradiation doses, 5 days consecutive at 2 grays per day. And in this case, you can see, yes, this is a combination treatment. So you can see here that when we only irradiate the tumors, the T over C is about 50%, and when we add a compound, we can decrease the T over C to 25%. So about compounds, I will not name the compounds because these are confidential. So I will just put compound on compound 1, and you have to know that all these compounds have been involved into inhibition of apoptosis or DNA repair. Another experiment with a combined treatment. In this case, we use LOVO, which is a human colon tumor. In this case, we irradiate, we perform a standard irradiation. And here you can see the vehicle, and you can see here the only irradiation in empty squares. And here is the compound at the highest dose. So you have it has a T over C. It has a significant effect, quite similar to the irradiation alone. And if we use this dose free with irradiation, then we induce a lethality, a big lethality. So we characterize where was coming this lethality, and we observe that this was not due to irradiation by itself, but it was due to anesthesia by ketamine xylazine that induced the lethality. And at a lower dose, it was working efficiently. So if you take the dose, the first lowest dose, then with TLF30, there was no significant effect, but at the intermediate dose, there was a T over C of 35%, which shows an increase of irradiation effect. So now I will describe you the in vitro assays for radiotherapy. So all of these are performed in clonogenic assays. And these are standard tests for evaluation of chemoradiation and radiosensitivity by opposition to MTS metabolic tests that are commonly used for cytotoxic assays. The main reason is that irradiation defect might need several cycles of DNA synthesis that might not be feasible in MTS assays. So how do we proceed? We seed the cells in two different methods. When we put 2D monolayer assays, they are in liquid, so they are sitting at the bottom of the plate. And we can also perform anchorage-dependent assays, which are performed with agar. And in this case, they are in agar and growing in 3D. So 1 hour after, we irradiate the plates. And after 1 to 3 weeks, depending on the cells, we count the clone at the bottom of the plate in case of 2D, and inside the agar for 3D experiments. And after, we have to evaluate what is the activity of the combination. So there are 3 different activities that can be reached. So only additivity, so it means that the treatments are acting independently from the other, and the combined activity is the sum of both treatments, the addition of both treatments. And they can synergize, and then the treatments are acting together to potentialize the global effect. And the combined activity is higher than the sum of both treatments, and also antagonize. And in this case, one treatment inhibits the activity of the other one. The combined activity is then lower than the sum of both treatments. And to classify the activity of this combination, there are there are many different methods of calculation, and here I report 3 different ones. First one is additivity method, which is also called combination index calculation, and this is a simple model of calculation because 1 irradiation dose is enough to make this kind of calculation. There is the classical Schutelele method, which is often used for combination with two chemicals. And this is a more complex method of calculation, which needs an algorithm and use of a software. And in this case, a multiple irradiation dose is needed. And the last method I will show you is the SER method calculation, which is a sensitizing enhancement ratio, calculation of this ratio. And it's a curve comparison. And it needs also a multiple irradiation. So for combined treatments, in vitro, I will first show you an example performed with human head and neck FADU and human U87MG glioma. And this treatment, we irradiate in combination with anti-DNA repair inhibitors. The assay was performed in monolayer, and we count the clones about 2 to 3 weeks of incubation with compounds. So as you can see, for U87, even at the lowest dose, there is an effect of the inhibitor. At the lowest dose of irradiation, there is an effect of inhibitor for both inhibitors, indeed, because we are using inhibitor 2 and inhibitor 1. For FADU, the effect of inhibitor is not there is no effect of irradiation at the lowest dose of inhibitor. So how do we calculate the index combination? So we have to calculate first predicted theoretical cell viability, which is obtained by multiplying the irradiation and the compound activity of treatment, single treatment, and is corrected by 0.01 to have it in percentage. And with this predicted cell viability index number and the measure one, which is measured on the plate, we can calculate the combination index with the ratio between the two values. And here you see, according to the value of the combination index, we can speculate about the in vitro combination activity. So if it's between 0.8 and 0.2 and 1.2, there is only additivity. If it's less by 0.8, then we start to have synergy, and if it's higher than 1.2, we start to have an antagonism. So here on the previous curves that I showed you, we can calculate the combination index at the different compound concentration and for the different cell lines and compound treatments. And we could say that, well, for when compound 2 is additive for U87MG, and for all other combinations, we have seems to synergize. So here I report because it was performed with the same compounds and the same using the same study, the in vivo studies that were performed with a combination of this compound 1 and 2 and in FADU tumor models. So this was performed with a standard irradiation process at 5 consecutive days at 2 grays per day, and with compounds orally given once every day for 2 weeks at 2 different doses. So we have quite the similar results for both compounds, and you can see here the vehicle, and after the compounds at the 2 doses are not really active, and this is TLF30 at TLF30, which is active given a T/C of 30%. And when we add the compound to TLF30, so this is a combination, then we had no significant increase of tumor regression. So now I will speak about the SCR method for combination analysis. And for this, I will show you this experiment, which was performed with different cell lines, the FADU cell lines, the tongue carcinoma Cal27 and SSC15, and the human cervical KB. And in this case, we perform a multiple irradiation at 0.5, 1.5, and 2.5 grades, and in combination with a single inhibitor that is an anti-apoptotic inhibitor. And the assays were performed in monolayer clonogenic assays. So here I reported the best responder to the molecules, and for this, you can see here the different irradiations that were performed, and in non-irradiated conditions, the compound is active with an IC50 of about 150 nanomolar, and a global diminution of cells surviving is observed following a dose response for all concentration of compounds. So here you can see the same curves obtained with the different cell lines, and you can see that Cal27 is a more sensitive cell line, followed by SCC15, KB, and FADU is not very sensitive. Maybe at the last dose it is more sensitive. So how do we calculate the SCR? So the SCR is calculated by tracing the line for the different concentration of compounds according to the different radiations. So when we trace, we take the area under the curve of this line, and we perform this for the untreated control, and we do this for the treated cells. And the SCR corresponds to the ratio between this area under the curve, which is the mean inactivation dose, and this is SCR, so is a ratio between the mean inactivation dose under untreated controls over the mean inactivation dose over after compound treatment. So here you can see what were the results after the calculation of this SCR. So in this case, we have the same numbers here, 0.8 and 1.2, but the synergy is characterized when we have a score over 1.2, while antagonism is when we have a score lower than 0.8. So we are in the reverse situation compared to combination index. And in this case, you can see that the Cal27 the compound is synergic in Cal27 cell line, is additive in the FADU and KB cell lines, and close to additivity, but with a little antagonism for the cavity line. So in conclusion, I presented you different ways we characterize in vivo and in vitro the combination of radiotherapy with other kind of treatment. Here I show you only chemical, but there are other things that are still possible to do. And what we observe here is that there is an increased interest for radiotherapy sensitizer because we have an increasing number of in vitro and in vivo studies. We have, I described you several protocols and methods, but we have other models that are available for in vitro and in vivo studies. So I think that through this presentation we can discuss about new designs and eventually characterization of new models of new studies if interested. So thank you very much to listen to this presentation, and I am waiting for your question now.
Thank you, Guillaume. For any attendees who have questions, please go ahead and type those into the Q&A box now. We have received a few questions. Guillaume, this first question is about controls. How are the non-irradiated controls processed during irradiation?
So indeed, what we try to do is to process them similarly to the irradiated conditions. So they are treated similarly for animals. They are transported and they are anesthetized as we do for irradiated animals to have exactly the same condition of experimental conditions. And for plates, similarly, we transport the plates in the same condition as the irradiated ones to have the same temperature variation or anything else.
Okay. The next question is kind of in the same lines. So how do you proceed for the sequence of administration of irradiation and the compound?
So for the sequence of treatments, we can have different cases presented. The treatment, but often in the experiment that I described you, they were performed quasi simultaneously. So what we do is that we treat animals 1 hour before and after irradiation. It depends on the mode of administration and can be decided protocol to protocols. And plates are treated before for agar clones are treated with the compounds before for agar clones, and also for 2D assay. But for 2D assay, we can think about just delaying the chemical treatments after irradiation with a concentrated solution that we can put into the plate after.
Okay. In the different examples of the irradiation models, the tumor growth inhibition was rarely less than 50%. Is this common for all tumors, or do you have some examples of higher activity?
So, well, what we do is that we use this daily treatment, 5 days, or even we can do 2 times 5 days treatment, and we use these 2 grays doses to be the closest to the clinical treatments for translational consent. So we can increase the activity by increasing doses, but it will not be we will not be in standard irradiation doses. So it will it's good to be at 2 grays. But also for combination studies, it's good to not be at 2 to efficient tumor growth inhibitions to see a combination effect. And I would say that we had some examples that I didn't put here where we don't see any effect of radiation by itself, and treatment with other compounds that are not really effective by themselves, but when we combine both, you can have a tumor growth inhibition of combination only.
Okay. Thank you. That is all the questions that we have received. So I want to thank everyone for attending the seminar. Thank you, Guillaume, for a very good seminar. And we will send out a link to the presentation shortly. Thank you all.
Okay. Thank you all.